Imagine walking into a hospital with a severe infection. Your doctor needs to know exactly which bacterium is making you sick. But there are billions of bacteria in the world, and many can cause similar symptoms. Waiting several days for laboratory results could delay treatment. So how do scientists quickly identify the likely culprit? The answer lies in one of the most important techniques in microbiology, gram staining. In less than 10 minutes, this simple laboratory procedure separates bacteria into two major groups and provides valuable clues that guide diagnosis and treatment. Gram staining was developed in 1880 by Danish scientist Hans Christian Gram. Although he simply wanted to improve the visibility of bacteria in infected tissues, his discovery revolutionized microbiology and remains one of the most widely used laboratory techniques today. Every day, hospitals around the world use gram staining as one of the first laboratory tests. Within minutes, it helps identify whether bacteria are present and provides essential information that supports early treatment while more detailed tests are still in progress. A gram stain reveals much more than the presence of bacteria. It shows whether they are gram-positive or gram-negative, their shape, and even how they are arranged. These details help microbiologists narrow down the identity of the organism. Before performing the procedure, the laboratory scientist prepares all the necessary materials, including a clean glass slide, inoculating loop, crystal violet, grams iodine, alcohol, safranin, distilled, water, and a microscope. The first step is preparing a thin bacterial smear on a clean glass slide. A small amount of bacteria is spread evenly and allowed to air dry completely before any staining begins. After the smear dries, the slide is gently passed through a flame. This process, called heat fixation, kills the bacteria, preserves their shape, and firmly attaches them to the slide. Next, the smear is flooded with crystal violet, the primary stain, for 60 seconds and rinsed gently with water. At this stage, every bacterial cell absorbs the dye and appears deep purple under the microscope. Grams iodine is then added and left for 60 seconds before gently rinsing with water. It acts as a mordant by combining with crystal violet to form a larger complex that becomes trapped inside certain bacterial cells. Now comes the most critical step. Alcohol is carefully applied for 10-20 seconds and then rinsed immediately with water to remove the stain from gram-negative bacteria, while gram-positive bacteria retain the purple color. Timing during this step is extremely important. Safranin is now applied as the counterstain and left for 30 seconds before rinsing with water. The previously colorless gram-negative bacteria absorb the pink stain, while gram-positive bacteria remain purple. The stain slide is examined using the oil immersion lens of a microscope. Purple bacteria are identified as gram-positive, while pink bacteria are gram-negative. Gram-positive bacteria have a thick peptidoglycan cell wall. During decolorization, this thick layer traps the crystal violet-iodine complex, preventing it from washing away. Gram-negative bacteria have a thinner peptidoglycan layer and an outer membrane. Alcohol disrupts this outer membrane, allowing the purple dye to escape before the cells take up the pink safranin stain. When compared side by side, the differences become obvious. Gram-positive bacteria appear purple with thick cell walls, while gram-negative bacteria appear pink with thinner walls and an outer membrane. Several errors can produce misleading results. A smear that is too thick, overheating during heat fixation, using old bacterial cultures, or incorrect decolorization can all lead to inaccurate gram stain results. Despite being developed over 100 years ago, gram staining remains one of the fastest and most valuable diagnostic tools in microbiology. It provides immediate information that supports patient care, disease diagnosis, and bacterial identification. Gram staining is more than a laboratory exercise. It is the first step toward understanding bacterial identity and selecting appropriate treatment. Every purple and pink cell tells an important scientific story. If you found this lesson helpful, remember to like this video, subscribe to the channel, and join us for more exciting microbiology lessons.